We talked about humans in the session before, and now we move towards the animal kingdom. And we start with Sevim Isparta from the University of Bochum. It's yours.
Thank you so much. First of all, I would like to thank the organizing committee for giving this excellent chance to me. Unfortunately, I couldn't join you in person, but I'm super happy to be here. Today, I'm going to talk about some of my preliminary results of my PhD project with you. Since this is a Laterality Congress, I'm not going to define any general terms, and we all know that hemispheric asymmetries are not unique for only human beings, and it's the common vertebrate feature, and the most apparent functional expression of cerebral asymmetry is handedness in humans, paw preferences, pawedness, limb preferences in animals. Since I'm working with rats, what about rats? Actually, investigating limb preference in rodents has a long history in laterality research. To the best of my knowledge, the first study has been conducted in 1930 with 105 rats, and this study has shown that females show more left bias than the males. And of course, the number of conflicting results has increased with the subsequent studies in rodents at the population level. And some of the authors suggested that they have leftward bias at the population level, while the others shown that they have rightward bias. And a recent meta-analysis conducted by our colleagues has shown that rats show individual level asymmetry, like being lefty or righty, but they don't show population level asymmetry, unlike humans. Why it is so important to assess behavioral asymmetry is because many studies, different studies suggested that this having lateralized brain has an evolutionary advantage. It was already mentioned in the previous talks. For example, lateralized periods and chicks were found more successful than the ambilateral individuals at cognitive tests, and also we showed that lateralized cats were more successful than the non-lateralized ones in problem-solving tests. For a long time, it was assumed that functional cerebral asymmetries are often directed by the genetic factors and they are constant over time, but it also has been shown that FCA has some degree of plasticity, and stress exposure may change functional cerebral asymmetry with different mechanisms. And one of the most known theories about that is stress exposure often needs more activation of the right hemisphere and result leftward behavioral asymmetry. And increased stress situations would lead to increased glucocorticoid levels, which are playing an important role in corpus callosum for interhemispheric communication and cause atypical asymmetries. These atypical altered asymmetries are known to be also associated with many different psychiatric disorders like depression, bipolar disorder, anxiety disorder. And the hypothesis of my study is prenatal stress alters functional cerebral asymmetry in rats. Within this scope, we are trying to address those following questions. The first one, is there any effect of prenatal stress on motor laterality in rats at different postnatal developmental periods? Is there any sex effect? And also, does prenatal stress asymmetrically influence the expression levels of some genes in between two hemispheres? This is the first study to investigate the effect of prenatal stress on FCA and its molecular background by using different behavioral tests and also genetic analysis in rats. Within the scope, I have two different groups, stress and control groups. And these groups consisted of the pregnant moms and their offsprings. As a prenatal stress protocol, we used restriction stress 2 hours every day during their last trimester of their gestational period. And from each mom, like the offsprings obtained from each mom were equally distributed to 4 different age groups, which are postnatal day 21, 40, 60, and 90. And each of the animal was tested for only one age group. And after the testing phase was completed, they were sacrificed to get their brain tissues. As a different behavioral test, we designed a new testing apparatus by combining 3 different motor laterality tests, which are pasta matrix reaching test, Collins paw preference test, and head turning asymmetry test. Actually, this testing apparatus allowed us to reduce the habitation period for the animals for new testing setup, because we know that rats are neophobic creatures and it really takes a lot of time to habituate them to the new apparatus. And also, since this apparatus is adjustable, it also allows us to test them at different developmental stage, regardless of their body size differences. And this is the Collins test. Most probably, many of you already know, this is the most standardized methodology to assess paw preferences in rodents. And animals needs to use their paws to reach the food through the tube. And as a reward, we use the commercial Cheetos brand, and they really like that. And when the animals reach 50 paw interventions, we terminated the test sessions, and each test session was 30 minutes. And some of the animals reached the 50 paw responses in a single testing days. Some of them needed some more days. And after the Collins test, we also performed pasta matrix test as a second limb preference test. The principle of this test is also same. It is a foot reaching based test. The animals needs to use the paw. Again, we tested them until they reach the 50 paw responses. You can see uncooked pasta here, and we also dipped them to the hazelnut cream to make it more desirable for them, because it's sometimes not easy to motivate them for testing, because we don't restrict them for any food or beverage. And the last motor laterality test is head turning asymmetry test. In this test, we use 50% hazelnut syrup with sugar to assess their natural drinking behavior. And differently from the original paper, we didn't restrict them from the water, because we don't want to test them under any negative situation. And the test perfectly worked, and the drinking duration was much higher in comparison to original paper. And this test also conducted 4 consecutive days. Each test session was 30 minutes. Like a human research, we also use laterality index and binomial z-score. Laterality index is used for determine strength of laterality, and binomial z-score is used for determining the direction. When the animal has positive z-score value, they were classified as right paw. When they have negative z-score value, they were scored as left paw. And I would like to show you some Collins preliminary results. In line with the meta-analysis, in our study also showed that they show individual level asymmetry in both of the groups like stress and control, but they didn't show population level asymmetry. And I think it is the interesting finding of our study, because the first paw intervention might be used as an indicator of general paw preference for direction. And this is really promising results that can be used for further studies. Maybe we don't need to test them until they reach the 50, but of course we need more animals to confirm this conclusion. And to investigate stress, age, and sex effect, we checked stress, age, and sex effect for absolute laterality index between groups, but we didn't find any significant differences. But when we checked the interaction between stress and age groups, again for absolute laterality index, we found a significant interaction in females. As you can see from the graph, the control group is green and stress group is red, and less laterality in the stress group increased with age. But when we checked males separately, interaction was not significant, but we should be careful here to interpret the results, because we had some troubles for P60 and P90 groups for males, because somehow we didn't motivate them enough, and the number of the animals who reached 50 paw responses or solving the test was really less than the other groups. Therefore, we are trying to increase the sample size now. And as a conclusion, first POP intervention can be indicator of general POP preferences for direction in rats, like cats and dogs, because we also showed that before. And prenatal stress may influence the development of lateralization in rats and result in reduced FCA. Sex hormones may be one of the additional factors which influence FCA in general. And as I said before, for further validation, we need larger sample size to confirm these conclusions. Shortly, I would like to also share some ongoing data analysis with you. We also performed elevated plus maze test to assess anxiety-related behaviors of the animals. And the first analysis, however, we didn't indicate any significant differences between stress and control groups, but we are still ongoing. And in addition to this, we are also analyzing the effect of prenatal stress on turning behavior because by using deep lab cut, because a very recent study conducted by our colleagues showed that early life stress leads to more left turns than rights in rats. But as I said, still we are working on it, and very similar to turning behavior, we are also using deep lab cut for head turning asymmetry test analysis. And we extracted 2 frames from each video. And the first model was created for, I think, more than 400 videos, 415. And we did manual labeling for 30 different body parts of the animals. But since the video size are so high, GPU really takes time to analyze the videos, but we are still working on. Hopefully in the next time, we will also share those results with you. As a future direction, our main aim to increase sample size, to complete behavioral data analysis, to choose best samples for genetic analysis, because from the selected brains, we will do DNA and RNA isolations. We already did the all optimization periods from the motor cortex, and we will do red gene expression arrays, which allows us to investigate the expression levels of more than 30,000 genes at once. And if you find any significant differences between hemispheres, we will do qPCR confirmation validation for these results. And actually, it is also one of the benefits of working with animals, because there is no study can be conducted in humans with these brain tissues. I would like to thank to all my colleagues and supervisors who helped and support me for this study. I'm happy to try to answer and your questions, and yeah, I'm waiting for your comments.
Thank you very much, Sevim. Now, can you hear me?
Yeah, I can hear you.
Super. Then questions.
Hello, and thank you very much for this very endearing and wonderful talk with very interesting results. I have worked with a student also on this POP preference in rats, and as we were going through it, I was wondering also about the ecological relevance of a reaching task for rodents, just in terms of general food acquisition. Like I can imagine for cats, it makes sense to have a paw preference. For dogs also, because they really use them in food acquisition. But for rodents, I'm not quite sure if paw preference is the best, the best behavior to see a laterality in. Have you considered also looking at what you did a bit with the head turning at whisker preference, for example?
Actually, it's a good question, because we are always discussing about the ecological, from the ecological perspective, because as you said, for example, dogs looks like use more paws than the rodents, but actually not, because they are using their paws for digging behavior. In my personal opinion, when you compare with rats are really using their paws with really good motor ability because they are reaching the food, holding the food, and also when you look at the original paper of spaghetti test, you can see like how they hold, and there are different measurements how they use their pose, like how much manipulatively. To be honest, for rats, I think it's a good indicator to assess behavioral laterality. For the whiskers, I really don't know how to measure because most probably we need really high resolution of cameras, maybe more than GoPros. Because even with paw preference, while we are watching the videos, sometimes it is not easy to count their paw preferences because sometimes they are using real paw interventions, sometimes like little. But I don't know how can we measure, but of course, why we don't try to also maybe licking behavior and whisker behavior, and also depending on the stimulus, we can also maybe check turning. And yeah, I'm not sure I could answer your question perfectly, but
You get thumbs up from me there. It was a good answer. Now there's a question coming from Tom.
Hi, thank you. It's still interesting, all this plasticity in both pathways. I was wondering, so I suppose you think that these effects are mediated via steroids. And I was wondering, is there any evidence for an asymmetry in receptor distribution for these hormones over the two hemispheres?
I'm not sure. Could I hear you perfectly? Could you please repeat your questions because it comes from really deeply?
Oh, okay. So do you know how this early stress affects laterality? So is there any evidence that it is going via steroid hormones? And if so, whether there is an asymmetrical distribution of their receptors in both hemispheres?
Yeah, I could hear now. I think there are some studies to assess prenatal stress effect on FCA, but they are really old studies. I just could see like 3 of the studies, but all of them were a behavioral level, and they didn't check any molecular backgrounds like receptor based on the effect. Therefore, I cannot tell you. Maybe there is some papers I didn't catch before. Yeah, just they saw the differences, like they said functional cerebral asymmetries can be reduced with the stress. There are some, for example, one of the hypotheses is because we are exposing them the stress at their last trimester of their pregnancy because of the motor cortex developments, and they said, for example, testosterone levels are making a peak, like a peak highest average in the last trimester, and maybe we are with the stress exposure we are destroying that testosterone levels at that time period, and therefore maybe their brains are not developed as they should be. But this is one of the hypotheses from one really old paper, but I don't know the other papers or in the literature.
Okay, thank you.
Okay, and surely a quick one from Marcus.
Hi, Sevim. Thanks so much for your very nice presentation. I have 2 questions. Sorry. So one is, you mentioned there's a significant interaction for women, but there's no for females, and no significant interaction for males. Does that mean there's a significant three-way interaction? And second, you've mentioned steroid hormones. And I think my second question is very much related to the previous question from Tom. So how do you think stress hormones, or I should actually say sex steroids, will actually interact with stress hormone levels in your experiment?
I could hear your second question, but for the first one, if it is not a problem, I will ask you again because I couldn't hear, the voice was frozen. And for your second question, after we found these preliminary results, I checked the literature and there are many conflicting results about the stress hormones and their interaction with the laterality. Therefore, I think there is no really clear answer of this question. I really don't know, because maybe I shouldn't say like this, because there are many hypotheses like how they are interact with each other, like because of the menstrual cycle, the women have different laterality index depending on the times. But I really don't know how to interpret this interaction right now. Maybe if it is.
I think an honest answer is better than a wrong answer. So my first question related to the interaction, because you reported, there is a significant interaction for females, but no significant interaction for males, which suggests there is a significant three-way interaction. So was the three-way interaction significant or was it not? I think that's critical.
I still couldn't hear you. I apologize.
Did you have a three-way interaction?
A three-way, no, like sex, age, and stress, right?
Yeah.
No, we didn't find. When we checked the interaction between stress and age, that then we could find the interaction. When we did the three-way, we didn't find.
Okay. Okay, thank you very, very much. I'm sure we're going to hear much more about stress and laterality in the future. And we're certainly going to hear more from you in the future. Thank you.
Thank you, Sue.